And there are, you have short, long signals, and you have some that are act on the same cells, and so on ones that act on other cells. Okay, so now I'm going to explain each one of those. So Duncan was correct. So hormones are a perfect example of molecules that have a long range. So for example, right now, epinephrine that is produced close to your kidneys can be sent to different tissues, and if they have the correct receptor, they can increase your heart rate. Yeah, you can also have other ones that can reduce the heart rate. Yeah, but they need to have the receptor. And the most important about these ones, the hormones, is that they go through the blood, yeah, through the blood system, so to the circulatory system. So if you want to find hormones, they can be in many places, but definitely they will have to travel to the highway, and the best highway that have the body are the circulatory system. This is why when cancer goes into the circulatory system, you start getting into trouble because it can start traveling to any place. And the other one is called the local mediators. As you see, the cells have to be very close to have a response. So this one is called endocrine signals, and this one are called paracrine. The crine means secrete, yeah. So the paracrine, para is besides, and endocrine it will be that is secreted inside, that is sent to other sections. Okay, so remember, endocrine signals are large range signals that are carried through the bloodstream. Very important. While the other ones are the paracrine signals that are released locally. So you can divide this cell signaling by the distance, if they go far or they will go close. The other example is the cells that they act on, yeah? So you can also divide it in this way. So if you see here, I put a person that is poking the other one, so they are not talking, but they should have physical contact. You will find that some signaling that are very important are this one, that two proteins of a cell, they need to be in contact. And another ones that the same cell has to secrete their own signal, and it change it produce a response in the cells, and I will show you some examples. Okay, so this one is called juxtacrine signal that require a physical contact, and the other one, it will be autocrine signal that act on the same cells that produce them. Okay, so let's talk about more about hormones. The hormones are key for multicellular organism because you can send one hormone in one part of the body and regulate and coordinate the morphogenesis of different tissues that are far from each other. And it's not only important for animals. All of us know about hormones in animals, but in plants they are also very important, and they have been studied in a lot of detail. So again, for example, there in red you have the hormone A that is attaching to a cell, and the most important is that it has to have a receptor that have the same conformation, yeah, that there is a complementarity in the shape. Here, the cell, the hormone B is going to attach to the hormone B because it had also complementarity, and here these cells both can attach to this one, yeah. So that is very important. Now I'm going to show you an example of a hormone. As I told you before, in the adrenal glands, in the kidney, the epinephrine is secreted, go to the bloodstream, and they can go to different tissues. In many tissues, and most of the tissues, there will be no effect. But if they have these red spots, these green circles in the schematic represent the receptor, if they have that receptor, they will be able to have an effect to start the signaling. Yeah, they will form weak bonds, they will attach, and then the signal transduction it will start. And for example, in the heart, it will increase the heart rate, the epinephrine, and here in the liver, it will allow that the glycogen. So all this Angus, I forget what is glycogen. Could you help me? Could you remind me what is glycogen? In honor of John. John Brubaker will be very proud.
I don't know if I remember it after.
Yeah, try to. What do you think it is? Okay, glycogen. Just in case for the test, if he asks you glycogen, is when you have a lot of sugar stored and you have to break it into glucose. Good.
Oh right, yeah.
Asher, I'm lying. No, yes. For a second, I'm like, I'm confused. No,
I think you're good.
is it glucagon? Glucagon. Yes, I sometimes I just remember them in Spanish. But yeah, they did that. And in this breaking of the sugars, we will see it in another class. Right now, I'm just telling you there is a hormone that goes there. Later, we zoom into that and we see all the molecules that are involved in that breaking and that we go into a lot of detail. Okay, so far I told you that there are some type of signal that is called endocrine signals. That is very important that you think about the circulatory system. Also, I told you that the endocrine system is secreted directly through the bloodstream, so they take that highway. And I haven't told you this, that the lifespan can be from few seconds to many hours. Super important. What determines how long they will be attached to each other? The complementarity. Yeah, so you can form weak bonds, you can form strong bonds. It can be it's like the lock and the key that they always show you between substrate and enzyme, that metaphor. So same thing happened here. If the lock fit perfectly, the key will open the door. If the ligand perfectly bind to the receptor, they will make better bonds, okay? And there will be more affinity. And we will talk about that at the end of the class. And I also told you that they need to have specific tissues, they have specific receptors that allow you to target specific tissues. In this case, I show you the liver and I show you the heart. Okay, so Asher, super easy question.
Circulatory?
Yeah, so you can find it definitely in all of them, but the most likely to find it will be in the circulatory system. Yeah, why? Because all of them travel through there. Yeah, there could be some that go to each one of those, but definitely all of them have to go to the circulatory system. Okay, and this is correct. Okay, somebody that take anatomy and physiology, maybe I don't think that you will remember. You are not yet in that chapter.
No, I think it's D.
Is okay. How many people say D? All of you say D, and you are correct. So there are 4 categories. You have amino acid, derivatives, peptides, proteins, and lipid-like hormones such as steroids. And so, for example, the amino acid will be the epinephrine, the peptides will be vasopressin, the proteins will be the insulin, and lipid-like, there is a lot of them, and here we have this testosterone, okay? Okay, so Eric, excellent, and Enrico, good job. So in the quiz, this will be the correct one. Derek, sorry, Super easy question. Which of the following bits describe the autocrine signal?
B.
Signal that act on the same, yeah, so auto itself and crine that secretes, yeah? So pretty easy. So, oh, some of you have asked me not to scare you, is that, so some of these questions of the quiz are pretty easy. So in the final exam, I will reduce some of, I will still keep some, but there will be some more from the textbook. They will still be in the practice test, yeah. But just that you know, so there will be questions, easy questions like that, but there will be also more questions of the textbook that you can also find it there, and they will be, and you can read the textbook. I knew that this test is the one that you are tired the most and you have no time to study, so I didn't want it to make it more difficult for you. But the final exam will have a little bit more of complexity than this one. Not a lot, but just to remind you, not. Okay, the classification of molecules. So here, as Derek told us, there is autocrine signals that, as you see here, the cell will secrete signals and that it will receive himself. And here they show you 4 examples, so negative feedback, positive feedback, self-stimulation, and transactivation. So why they do this kind of thing? So for example, in the endothelia cells, for example, you are playing basketball, you're playing volleyball, you're running, so you need more blood, yeah? So if you need more blood, the endothelial cells are going to change. They said like we need more blood supply here, so they send a signal to increase the blood supply, yeah? That more blood go through this tissue. And if the opposite is happen, if you say, oh, I'm resting, I'm in the bench right now, I need to rest, so you can go in the opposite direction. Self-stimulation, here they gave you an example there, an inflammatory cell. So there is one cell, an antibody, yeah, and it encounter a bacteria, and it said, oh, we need to destroy this bacteria. What it does, it will produce, in this case, the IL-2 signal that it will receive, and it will allow him to proliferate, and when there are many, he can attack the bacteria. Okay, so here I show you one example, here I show you another example. This activation is, this could be also a paracrine. In this case, the fibroblast is receiving himself the signal. It could also receive a fibroblast that is close there. The idea is you need to produce a lot more of collagen. There was an wound, so in that case, having this signal allowed to produce more collagen. Okay, so in those 4 examples, we see a signal, a cell that is producing a signal that is allowing to change its behavior. Okay, and remember, this is like when you write a note to yourself, the cells are saying, we need more to proliferate more, we need to change this behavior. All the things are doing that. So perfect. Now the last signal is these juxtacrine signals that require physical contact. And here we start getting into a little bit more complex stuff. So here you see 2 cells. Here you see Notch. This is a protein that is super important in a lot of cell signals. Here you see a protease, and here you see that in the nucleus there is a repressor, so there is no transcription. Now, when there is, in the second example, you have the delta and Notch that the other cell have a physical contact. So what happens is when there is physical contact, this enzyme that the function is to cut a protein, it cut it, and now this one will have more affinity, yeah, this will travel through the nuclear pore to the nucleus, and it will have more affinity than the repressor, and it will start the transcription. Okay, so again, here juxtacrine the juxtacrine signal require physical contact. In this case, it was delta and Notch. and it leads to a series of events, a cascade of events that produce a response. In this case, it was a change in the gene expression. And this is a simple case. In the beginning of the class, I show you like a domino change reaction. You will see, this is the part that I don't like a lot when there is a lot of molecules involved, but you will see that I will try to walk you that that they will start getting more complex and more complex all the pathways. Okay, so we finished this, and I'm sure that you will remember that there are different type of signals, one that are depending on the distance. Yeah, and we talk about hormones and paracrine signals, and also depending on which cells they act. So we have the juxtacrine and the autocrine. Now we're going to talk about all these different components that you find here. Yeah, all these signal the perception, intracellular transduction, and cellular response. Okay. So the cells require a lot of signals. Yeah, and those signals are very important. And in this case, you will need a receptor to interpret that signal. That signal, if you see here, it will release different chemical signals, and in that way is that we control the different the development and the different activities of the cell. Okay, so I know you're very tired, so instead of me talking a lot with the person besides you, here are the keywords for the test, the basic ones, and here are the definitions. Yeah. You have 2 minutes, try to match them, and hopefully with this it will help you more than me talking for 20 minutes about each one of those, and I'm here to help you. You know? Yeah. Exactly. Chemistry, video class. So try to match that, and I think it's pretty easy. And you have the slides on, you have the answer. Oh, did I put the answers? No, that's Nicholas, he forgot. Okay, so the specific site on a receptor or the ligand binds, what is that?
The binding pocket.
Perfect.
Okay. So
binds from receptor to receive the signal.
not that. Okay.
Perfect.
Should we written like there?
Yeah.
Who's got what?
Or ligand, I don't know how to pronounce it. I'm sure that, let's pretend that we are British, Australian, so we can pronounce it.
I can never pronounce it.
I don't know, I think it's like, I have to written.
I've heard so many
Differences between primary secondary.
so many it's
No, see, that's what I was kind of
Okay, Alicia.
messed up on. So C and D.
Okay.
Primary.
It's a functional intracellular response. What is that? Perfect. Okay. Okay. Oh, you're also doing hundreds of things at the same time. My brain right now is thinking about different things. Okay. Easy. Okay. The secondary messenger and the primary messenger. What's confusing to me, but they are
Disgusting.
you have it here. Secondary messenger that goes using the cytoplasm, and the primary will be this one.
Okay.
Yes.
There you go.
Numbers of this.
Okay, so I tried this because I know that if at this point no coffee, I will have to give you in the beginning of the class instead of a quiz a Red Bull to all of you, and still we are too tired. So here we have all the concepts that we need: ligand, receptor, transduction, binding pocket, and I will keep the primary and second messenger for the next one. But the idea is that, oh, I gave you the first answer. The ligand, it will be the molecule that often, the molecule, often a hormone, a neurotransmitter that binds to a receptor. So it goes there to the receptor that in this case in the cell. Okay, I don't know, we were in Jana. I don't know if you write them down. Did you write it down? No. And I didn't give you the answer. So what is the receptor? The receptor will be the protein that had or a molecular structure that is in the cell surface. Yeah. The transduction is the process of converting the extracellular signal into a function in the intracellular response that it will be here. Here, the response is the outcome, that it could be changes in the gene expression, secretion, movement, or something else. Gianna,
Yes.
what is the binding pocket? This one. Don't worry, the binding pocket is this. Do you see the top of the receptor where the ligand is going to attach? So it's the molecular signal that initiates the entire signal process outside of the, oh no, give me a second.